I want to, Miles, I want to jump right into that polling question. We're 15 minutes into the conversation here. It's time for our first polling question. This question is for you, the audience. What form of vaccine or therapeutic are you developing? Your options are linear mRNA, circular mRNA, self-amplifying mRNA, oligo RNA, siRNA, other, or we are testing multiple formats. Take a minute, make a selection there, and we're going to jump back to those poll results in just a second and discuss them. In the meantime, we have a couple questions around chromatography coming in from the audience. Patrick, I'm going to jump back to you. One of them would be, maybe we can tackle these in tandem. What is the best practical chromatography for reducing double-stranded RNA? And then, what was the other chromatography question we saw there? Oh, yeah, talking about process time and yield, is there a time limit on column size? Or is there a limit on column size? Do people scale out and just have multiple chromatography systems? That question's from Ryan. The first one was from VJ. Patrick, you care to tackle those in tandem?
Yeah, principally, so best practical chromatography, there are some publications out that describe the process of chromatography for mRNA-based therapeutics. I think I would refer you to these. Maybe for your particular construct, you want to do some adjustments to this by, sometimes also depends on the process that you have before, so how pure your system is already. The other one was on scaling of chromatography. Mm-hmm. Here I have to admit that I'm not a complete expert because I'm more on the research side, so usually smaller volumes. I'm not so familiar with the end game of very large scales. So sorry for this.
Thank you, Patrick. Christian, anything to add to those two questions?
As we are HPLC free, I'm not too familiar with the limitations here. We deal with it in another way, as mentioned a second ago.
Okay. All right. Perfect. Let's take a look at those polling question results. Form of vaccine, therapeutic development, linear mRNA, 35.4%. We're testing multiple formats, 39.6%, and then other. So what do you think about that, Patrick and Christian? What are your gut reactions to this? Testing the bulk testing multiple formats. What's that? What does that tell you? What's that indicative of?
Yeah, principally it shows that it's still dependent on the type of application, maybe which format is the best. I think for some, even for vaccines, people are applying self-amplifying messenger RNA or linear RNA. I think all of these different sources have different flavors or different, also again, edges and so on. So self-amplifying messenger RNA has the opportunity to prolong the pharmacokinetic profile of the expression. So you can have longer expression, longer exposure of your protein, but it comes on the cost that you have very large constructs that bring some pressure in terms of production and stability. But also you have the non-structural proteins that come with the replicon. And therefore, these foreign proteins can also have some problems maybe outside of the vaccine world, yeah, because you can raise immune responses against them. Linear messenger RNA, I think, is currently also in this poll the most advanced thing. Yeah, it's best understood. There's a lot of publications out there. We have 2 products on the market. Circular RNA is a new player in the field. I think here it's also preventive a little bit that the production is not completely controllable or is not as easy like for a messenger RNA. So there is some technical obstacle to use this. But I think in the future, with all the people now coming into the field and having different applications, we will see that each company or each player will find the perfect solution for the task that they tackle. So I would assume that we have in the future all different modalities on the market, circular RNA as well as self-amplifying as well as messenger.
Mm-hmm. Yep. Christian, from the therapeutic point of view, is there anything that jumps out at you about this response?
I would fully agree with Patrick. All of them have their relevance, and all of them bear their opportunity, because all of them have a certain advantage, as outlined by Patrick already. Not going to repeat what he was saying, but I fully agree. There might be different indications that have different limitations, and therefore the solution might be another one, dependent on the target which is addressed and on the disease which is planned to be treated, and therefore all of them will have their relevance in future products, I'm sure.